I like this picture in your book. It shows you the systemic circuit starting at the heart, and it shows you the sequence of blood vessels as you travel from the heart and back again, which of course I don't have the left side of the diagram. I just have the arteries listed here. If I were to give you a question on an exam that said, order these vessels in the order that you would encounter them from the heart and back again in the systemic circuit, you would say, oh, first I would encounter elastic arteries, then I would encounter muscular arteries, and then arterioles. And although I've included them here, capillaries are not arteries. I've just included them here because they're on this side of the diagram in your book. As you move from elastic to arterioles, obviously the diameter becomes smaller, but you also have less elasticity. So there's less ability to stretch, which means that as we go from elastic to arterial, the main purpose of an arteriole is to contract or vasoconstrict. And I'm going to say this here, and I'm going to mention it a million times again. I used to teach from another book, and they did not call these elastic arteries. That's a name that refers to the anatomy of the arteries, which is current and correct. The old book that I taught from called these conducting arteries, which is true. They conduct blood to very general parts of the body, up, down, arm, side, side. Elastic arteries, like the aorta, have an incredibly thick wall, especially those like the aorta that are near the heart. And this is because as the heart pumps, it increases pressure to 120. And these arteries near the heart don't want to have like a blowout. They have to be able to take that pressure. So you can't constrict these arteries, which is strange because you're like, why is there so much tunica media, right? Why is there so much smooth muscle here if I can't constrict them? Well, these arteries, although they don't get smaller, reducing their diameter, they can control or keep their shape. So they can't vasoconstrict or get smaller, but they can recoil as the heart lowers or stops pumping and lowers its pressure to 80. These vessels kind of recoil and fill again. As these vessels rebound, they push back against the heart. There's a valve in the heart called the aortic valve. That's what controls blood leaving the heart to the body. And elastic arteries like your aorta pushes back on this valve for a split second. And that is what's called afterload. It's the pushback. We'll talk about that more specifically, especially in the blood pressure video. The other thing is that you have vessels on the outside of the heart. Let me draw a diagram of the heart for you. Oh, that's not anatomically correct, but you do have vessels on the outside of the heart called the coronary circuit. And these are the vessels that serve the heart muscle. And if these become blocked, you can have a heart attack as your heart muscle gets deprived of oxygen. These vessels, as the heart contracts, they actually get occluded or they close up. So as the heart rebounds, blood gets pushed into this coronary circuit. So for a split second, the vessels on the outside of your heart don't get any blood flow. Okay, so it's important for us to realize that these don't vasoconstrict. So in a way, knowing that, we kind of know that they don't really control blood pressure. These are muscular arteries. The other book that I used to teach from called them distributing because they distributed blood to specific organs. So the renal artery that comes off of the aorta and brings blood to the kidney would be a distributing artery. The term muscular artery refers to its anatomy and the fact that it has a lot of smooth muscle. You can see the internal elastic lamina on this picture here on the left, and you can see the external elastic lamina. Distributing arteries, their big defining characteristic is that they have a lot of smooth muscle. So they can vasoconstrict. And you might say to yourself, oh, maybe they control blood pressure. No, they don't. The vessels that control your blood pressure, that dictates the 120/80, those vessels are called arterioles. And you might say to yourself, no way. They're so tiny. There's no way that they control blood pressure. They do, because there's so many of them. Although their diameter is so small, their collective diameter is huge. So here's one arteriole. Here are 5 arterioles. Take the collective diameter, add up all of the diameters, and arterioles have more surface area than any other vessels. So it's these that control blood pressure. And this is why I liked the book that I taught from, because it called these resistance vessels, telling you that these are the ones that resist blood flow the most. And these are the ones that contribute the most to blood pressure. These have a pretty thin tunica media. They don't have a lot of smooth muscle compared to their diameter. However, again, their collective tunica media is what's important. In fact, look at this picture down here on the left. You can see that the tunica media, those sheets of smooth muscle that I'm highlighting here, those sheets of smooth muscle, they're not even like full sheets. You can see the endothelium peeking out from between them. So that's like a sweater with holes in it. This is the thing, though, is that the diameter of these arterioles, it determines the blood flow to any of your tissues. At the termination of this, remember that first diagram from this video, at the end of an arteriole is a capillary bed. And that's where your blood flows into the capillary bed, and all of the substances like glucose and oxygen leave the capillaries into your tissues. Arterioles determine the flow to a capillary bed. So when you exercise, your arterioles kind of shut off the flow to your stomach and digestive system, right? You're exercising. You shouldn't be digesting your food. And this is why you shouldn't go swimming for at least 20 minutes after eating. Well, it's true. All right, capillaries are the smallest of the vessels. They have the smallest diameter. And we know that capillaries can be thin-walled and fragile. They are. They only have a tunica interna, no tunica media, no tunica externa. And that means that they can blow out from pressure. The pressure in capillary beds is quite low. But if you have pretty high blood pressure, you can be squeezing too much blood into a capillary bed and blow it out. Some people will get broken capillaries around the eyes as they stay open for too long, as if you're studying late into the night, that can happen. This picture up here on the right is a picture of a capillary bed, an actual picture. So you can see the arteriole flowing right down the middle of that picture. Blood is just shunting down that arteriole, and then it's like spreading out into the capillaries. Down here on the bottom right, you can see that a capillary is just big enough to accommodate a red blood cell. The diameter of a capillary is just about that of a red blood cell. Now, if you have sickle cell anemia, your red blood cells get all clumped and clogged into capillaries, thus causing many of your symptoms. Capillaries, their purpose is to permit nutrient and gas exchange, right? We said that this is the point at which oxygen is getting off into the tissues and waste products are getting on into the capillary. So you want these capillary walls here to be extraordinarily thin, just one layer of squamous epithelial tissue. So it's nice and thin, and oxygen, carbon dioxide, glucose, hormones, they don't have to travel very far to get into what here is skeletal muscle. Capillaries don't work alone. They are found in networks or beds. This picture down here on the left, we'll look at this more specifically, but this picture, both of them show a capillary bed. One has blood flowing through it, and one has blood bypassing it. Let's talk about some little bits of smooth muscle that are called precapillary sphincters. Before we do that, I want to show you this capillary bed down here. And that this capillary bed, there's blood flow, blood coming up the arterioles, flowing through the capillary bed, and then leaving through the venules. So blood flow here is clockwise from an artery to a vein. Now, you can see here that these precapillary sphincters, which are just these little bits of smooth muscle, that they're only on the arterial side of the bed. Yes. And these precapillary sphincters in this picture are closed. And that's why, look at the capillary bed. It's not full of blood. It doesn't have its purple characteristic color, indicating that the vessels are full and that they're exchanging with the tissues around it. So this might be a capillary bed in your digestive organs after when you're exercising, right? Blood is just shunting through the digestive organ. It's just like, no, we're not going to slow down and drop off the kids. We're just going, right? Because we're not going to digest as we're running a marathon. The reality, though, is that you might say, oh, then if I'm digesting my food, my precapillary sphincters will be open. Yeah, kind of. Actually, they'll be opening and closing. So they're always opening and closing when you're exchanging items with the nutrients and oxygen with the tissues around you. And this is what creates a pulsatile flow. So this picture here is a picture of a capillary bed that is exchanging with the tissues around it. And what happens is a red blood cell comes up the arterial, leaves into this little tiny capillary here, and then it goes it has this pulsatile flow through the capillary bed, and then it starts flowing out through the venule. And this is so the oxygen and carbon dioxide can be exchanged. Think about it as a train. It's making stops along the way, right? It's not just shooting through all of the stations. This is actually a very good diagram in your book. Nobody likes it, but it shows you something that you already know. Let's look at this picture here on the left. This is a river, and this river is flowing down this tract of it. I gotta get a color that you can see. Let me do red. This river is flowing very, very fast, and then it hits the delta. And the delta has all of these little tributaries that fan out from it. And the blood is going really fast through that, the water is going really fast through that river there, but then it slows down as it hits the delta. And this is what happens in your capillary beds. Blood is flowing really fast, and then it slows down into capillary beds. And that's what this diagram down here is showing you, velocity, which means speed. Blood is going really fast in the arteries, and then it slows down. And then the speed picks up a little bit in veins, but it never really gets back to what it was in the arteries. Look also at the detail of this diagram, these graphs down here. Look at how the background of the graph shows you a red area that would be the arterioles. And you've got a little bit of purple, and then you've got the blue over here. And look at how this corresponds to the picture on the very top of this diagram, that it shows you a network leading to capillary beds and coming away from the capillary beds. Okay, so we get that, right? Blood flows really quickly to it, and then it slows down as it hits all of those capillaries. So the thing is, though, like, again, we might think of capillaries having a very small diameter, which each individual capillary does. But all of the capillaries together in a capillary bed actually have a combined diameter that's quite large. So I'm not talking about the diameter of one capillary here. I'm talking about the diameter of all of them combined. And that's quite large. And that's what this graph is showing you here. This graph is saying that arterioles actually have pretty small surface area diameter. And because there are so many capillaries, capillaries actually have a very large combined surface area. And then we also know that blood flow goes really fast until we hit the capillaries, and then it slows down. I'm sorry, has anybody ever seen that movie from the 80s, Better Off Dead? And he goes, just snorted. His friend is telling him how to ski the mountain. He goes, go that way, really fast. If something gets in your way, turn. There are 3 types of capillaries. They can be called different things depending on the book that you're reading from. All books agree on the fact that these are continuous capillaries here, and they're the most common. They're also the least permeable. Look at the cells in here, the squamous cells. They're all like right up against each other. There's only very, very small pathways between them to get into and out of the capillary. So these are continuous. They're the least permeable, the most common. There are fenestrated capillaries, and we'll see these again when we discuss the kidney. A fenestrated capillary, look at the cells on the outside of it, they're a little bit further apart. There are larger holes in between these cells, and those holes are called fenestrae, which means window. The last type of capillary can be called something different depending on the book. I chose this picture because I like the picture. Your book calls these capillaries sinusoidal because the holes in between the cells are large sinuses. These are like leaky. In fact, these are so big that white blood cells can leave between these cells, between these clefts in between the cells. And so these are called sinusoidal. Sometimes other books call them discontinuous. I like sinusoidal. Think about the bone marrow. Its purpose is to make red and white blood cells, among other things. And those white blood cells are pretty big. They have to be able to leave the capillaries or enter the capillaries of the bone marrow. All right, I know. Veins. We're almost done. Veins go quickly. Remember, this diagram over here on the right, we're following it from the bottom up, right? We've just exchanged with capillary bed, and we're going back to the heart because veins drain. As the capillary beds, as the capillaries come together and leave the capillary bed, they form a venule. So that's the first vessel that blood would encounter when leaving a capillary bed would be the venules. And they're still pretty porous. Actually, venules are still exchanging with the tissue around them. They're not really capillaries. They're leaky. Venules then come together to form veins. And your book says there's medium-sized and large veins. Other books make different separations between them. It's all veins. Remember, this picture on the right, this is not what veins appear as. They're not perfectly round. In histology, their lumen, or their open space, appears collapsed, as in the picture down below. They have not a lot of smooth muscle. Some of the largest veins have almost no smooth muscle, so it's not proportionate like arteries. It's not like the largest veins have the same amount of smooth muscle as the largest arteries. That's not the case. Also, veins have incredibly low blood pressure. And in fact, when we take your blood pressure, we're taking your systemic arterial blood pressure. The blood pressure cuff will not detect the blood pressure in your veins. Veins also hold about 60% of the blood in your body, which is interesting because when veins hold a lot of your blood, they're not even full. I mean, we said that before in a previous slide, that veins, they're not even full. They're like half full most of the time. Veins have valves. And if you've spent a lifetime on your feet, like, oh, I don't know, nurses and teachers and retail workers, you can end up with what are called varicose veins, and we'll talk about exactly what those are. So veins have valves. They're infoldings of the tunica interna, and that's what you see right there in that histological picture there. Those are the infoldings. And what they do is they prevent backflow. So as blood flows up this leg here, right? Blood is flowing up against gravity. What happens is it flows up, but then it kind of like surges backwards for a moment. This picture up top, blood is flowing through here. Now, let's say you were to sit for a very long time, blood starts to pool, And these valves here, they close the vein and prevent backflow. So if you've spent a lifetime on your feet, these valves kind of blow out. They don't close fully anymore. And this allows blood to really pool in your lower extremities and can cause varicose veins. What's the solution to this? Wear compression socks now. Don't put in 20 years of working on your feet and then say, oh my gosh, I have varicose veins, I should wear compression socks. Also, if you want the surgery for varicose veins, they will require you to wear compression socks for a pretty long amount of time before you get eligible for the surgery. So how to prevent them? Wear compression socks now, especially if you're working on your feet. All right, so I said that the blood pressure in veins is quite low. And if you think about it, they're really far from the heart, right? Veins are pretty far from the pump that's creating the pressure. So then what does cause the pressure in your blood vessels? What does cause the pressure that can cause blood to flow through your veins? So it's not pulsatile, right? We're not being subjected to contraction, relaxation, contraction, relaxation. We're too far from the heart for that. Veins also have very low resistance, which means that they hardly have any blood pressure. Blood is flowing through them, and it's not really being resisted. So what does cause pressure differences to suck blood through your body? Well, your muscles, your skeletal muscles are always pumping. They always are. Even when you're not using them, they're having a low amount of contraction, and this causes pressure differences, sucking blood through your tissues. Every time you breathe, your diaphragm moves down, creating pressure differences in your lungs. And these pressure differences move air, but they also move blood. So just like we can use a turkey baster to create pressure to push fluid out, we can do the same thing, creating pressure in your veins to move blood along. Your smooth muscle that surrounds veins also contracts continuously, so it also forms a pump that pressurizes the blood, moving it along. I said that your veins hold 60% of your blood. They do. I like this pie chart here. You can see that pretty clearly, right? About 60% here is blue. That's veins. So veins, in an older book I taught from, the biggest veins were called capacitance vessels because they had capacity or they acted as a reservoir. So that's number one, to understand that a majority of your blood is in your veins. Now, let's say that you nick your femoral artery and you are bleeding out from your femoral artery and blood is just gushing out onto the pavement in front of you. What's going to happen is that your veins are going to be like, oh my gosh, there's not a lot of blood coming back from the tissues. Let's mobilize our capacity. Let's mobilize the reservoir. And the systemic veins, those big, large veins, they constrict as much as they can. It's an abnormal constriction. It's a reaction to losing blood volume. And what they do is they constrict so much that they push blood into your arteries, right? Blood gets pushed back to the heart, and thus the heart redistributes it into arteries, which, yeah, the blood then gets pumped out onto the ground in front of you. Yeah, I know. But this is how the body reacts. You lose blood volume, we're going to mobilize blood from the veins. It's not only from the veins that we can mobilize blood, specifically veins in these organs, liver, skin, and lungs. The veins in these organs tend to hold a lot of blood. They are the largest reservoirs of the vein reservoir. And we can mobilize these also and push their blood out into circulation. And this is why, in a very simple way, yes, if you are bleeding out onto the ground in front of you, or if you are internally bleeding into a pocket in your body, you can become pale as you suck all the blood from that reservoir of your skin.